Hello, everybody. My name is Katie Politi. Welcome to the SWOG Immunotherapeutics Committee meeting. I am co-chair of this committee together with Dr. Siwen Huli-Scoven, who is joining us via Zoom. Welcome to everybody who is here in the room, and welcome to the people who are joining us via Zoom as well. Thank you very much, all of you, for being here. I think we have an exciting set of presentations today. So to get started, I think I'm just going to go over a couple of updates about the SWOG Immunotherapeutics Committee. And so do we have the, just a second. Okay, great. Thank you. So first of all, this is our agenda for today. So one of the, right after I finish going over these slides, Siwen Huli-Scoven is going to present slides related to the IMatch pilot study. This is the kickoff meeting for the IMatch pilot. So we'll have plenty of time to talk about that, and also for any questions from the audience for that. Just a second, I'm getting a message that the Zoom audience cannot see the slides. Would it be possible to share the slides for them, please? Just a second, everybody on Zoom. We're remedying that. There we go. All right, you should be able to see the slides now. So then we have two presentations, one from Dr. Shirley Liu, who is going to tell us about computational modeling of response to immune checkpoint inhibitors. And then one from Dr. Sean Bendel, who's going to tell us about single cell-based immune monitoring for IO clinical trials. Next slide. Next. So what are some of the things that we have been working on in the SWOG IO committee recently? One of the main things we've been doing is working on the development of the IMatch study, Immunomatch, together with NCI, including the initial pilot study, which activated at the end of last week. So we're very excited to be able to tell you about that. Next. So yes, the protocol is activated. Yeah, now it is. Yep. Now, and so one of the things that we can do is now we can begin to think about next steps and engage a SWOG IMatch working group to think about additional studies to be incorporated into IMatch and start thinking about those for the future. One of our objectives is to facilitate IO trial design and inclusion of TM in immuno-oncology studies across diseases. So some of the things that we've been doing is we've been inviting disease committees, the translational medicine chairs and their IO representatives to the IO committee to speak at the monthly meetings and tell us about some of the work that we're doing. So we're rolling that out across various disease committees. So some of you may be invited to present. You're all invited to attend to present at those meetings. We also discuss studies, and here I just put some examples of some of the proposals that we have discussed at our monthly meetings, and we'll go over the proposal and then talk about the translational medicine that is included in the study and have really a brainstorming discussion about it and about how to approach it and what to do. So anytime someone has a study in development and would like to talk about it with the IO committee, this is something that we can do. It's really helpful at any stage of development of a study. I think one of the other things that has happened is the Immunotherapeutics Committee used to be a research support committee, and excitingly, it became a full research committee this past spring. So that's one of the recent developments. I think One of our other objectives is to develop collaborative projects to study immunotherapy resistance, and this is something that we've been thinking about and seeing whether we could leverage SWOG trials and other cooperative group trials to study resistance, and this is something that will be an ongoing and future focus of the committee. Our monthly meetings are on the first Friday of each month at 2:30 p.m. Central Time. We talk about initiatives, we provide updates on ongoing activities, and investigators can present trials and ideas for feedback. If you're interested in information being added to the distribution list for these meetings, please contact us and we can add you. Thank you very much. So next, it's my pleasure to introduce Dr. Siwen Hu-Liskoven, who is going to tell you about the IMATCH pilot study, the BICAZO study. Siwen?
Yeah, thank you, Katie. And good afternoon, everyone. Can someone put up the slides? Oh, thank you. Okay, and first of all, I want to welcome everyone to the Immunotherapeutics Committee meeting today. It is really my great pleasure on behalf of the NCI and SWOG IMATCH team to announce the official activation of the IMATCH pilot as 2101. As Katie indicated, it was activated last Friday. So it's a phase 2 study of combining cabozantinib and nivolumab in participants in patients with advanced solid tumors stratified by tumor biomarkers. Next slide, please. So in the past years, we have seen the success of checkpoint inhibitor-based immunotherapy across different disease types, prolonging patient survival. But we're also seeing failure of many phase 3 combination immunotherapy trials, not necessarily because of the failure of the drugs, but because of the diluted signal in a unselected patient population with heterogeneous immune resistant mechanisms. So this is on the left of this slide is really the current state. There's no selection of the patients, for these phase 3 trials. And you can see, it's very hard to fish out the signal, efficacy signal. So of course, on the right is the ideal state. We would like to have precision biomarkers for individual combination, individual regimen. But we're not there yet. It takes a lot of time and effort to develop these biomarkers. So for IMATCH, from IMATCH, we really want to propose to provide a framework using biomarkers to not to predict patient treatment efficacy, but to try to predict our group patients into different categories of immune resistant mechanisms, of similar kind of immune resistant mechanisms, and test combination therapies in these defined biological groups. So next slide, please. So in this initiative, IMATCH is really aimed to enhance signal seeking. It's not really a match. I would say it's probably SWORD is probably a better word for it. It's to enhance signal seeking and by providing this comprehensive biomarker framework to evaluate immunotherapy in these molecularly defined subgroups based on these biomarkers. So a patient will, on the left of the patient, when they enter this framework, they will be assigned to these different biological groups based on the biomarker. They will enter the central protocol first, and then they will be assigned to these defined biological groups. And there are individual therapeutic protocols, based on after this central protocol. And the patient will assign to a particular protocol based on their particular molecular profile. And in the end of the study, there will be retrospective studies it will be done to further define and optimize the central platform and help to identify treatment-specific biomarkers. So this has not done in the past, this kind of central biomarker framework. It's really nice that NCI decided to run this platform to help the further development of combination immunotherapy. So, and because this has not done in the past, there is no current structure to support it. So NCI decided to run a pilot trial to first of all test the hypothesis and then also the feasibility of such a approach. Next slide, please. So the goal of this IMATCH pilot is to explore the IMATCH concept in a prospective trial to test the feasibility of prospective biomarker stratification and to streamline the central protocol operational structure. So that's why we developed this as 2101, the Picasso trial for this purpose. Next slide, please. So in the pilot study, we're going to test cabozantinib, which is a anti-VEGF based TKI in combination with nivolumab, anti-PD-1 nivolumab. And this combination has already been FDA approved for patients with advanced renal cell carcinoma as first-line treatment. And then we hypothesized that this combination can reverse the immune suppressive microenvironment and synergize to each other and improve anti-tumor efficacy and also patient outcome across different tumor types. And in this study, we're going to take patients with advanced melanoma and head and neck squamous cell carcinoma after they have already progressed on anti-PD-1 based therapy. And we plan to prospectively stratify patients based on the biomarker and study mechanism of response and resistance in these biological subgroups. Next slide, please. So this is an overview of the study. Patients with melanoma and head and neck squamous cell carcinoma who have already progressed on anti-PD-1, they will enter this trial. And there is a mandatory biopsy or recent archival tissue is also okay. This is required, and the tissue will be sent to the IMATCH screening platform, and whole exome sequencing and nanostring-based gene expression profiling is going to be done. And there are two stages in the trial. In the first stage, after the tissue is submitted, then the patient can start treatment right away without waiting for the result of the assays. There is a temporary hold at the end of stage 1, and there will be a pre-planned feasibility analysis. If the study can pass the feasibility criteria, predefined feasibility criteria, then we'll enter to the second stage. And in this stage, patients, the difference between stage 1 and stage 2 is, in stage 2, patient will need to wait for the subgroup assignment before they can start treatment. And there is a possibility if their cohort, the cohort that their biological group belong to is already full, then there is a possibility they may not be assigned to a treatment group. Next slide, please. So this is more detailed instruction how to do step 1 and step 2. Next slide is step 2. We'll just go through that very quickly. And then the next slide is an overview. Can we go to the next slide? So this slide is an overview what steps the patient will need to go through during the trial. So on the left, during the screening, there are these steps: registration step 1 and submission of the tissue, in step 2, there's a waiting for the cohort assignment, and then after it's assigned, and in stage 1, there is no waiting. Patient go to registration step 2 and start treatment. So the treatment is on a 28-day cycle, and nivolumab will be given every 28 days, and cabozantinib is taken orally daily. There are pre-designed procedures, which is pretty standard for phase 2 trials. And on the top, these are the mandatory and optional tissue collection that's built into the study. Next slide, please. So the primary objective of the pilot study, the most important objective, of course, is the feasibility of this molecular characterization based on tumor mutational burden and gene expression profile. Here in this trial, we use the nanostring TIS score for patient stratification. And also we want to look at the efficacy of this combination across disease cohort and also across and within tumor biomarker subgroups. Next slide, please. And there are also secondary objectives to further looking into the efficacy and the clinical outcome, clinical benefit of this combination in each disease groups, stratified by the tumor biomarkers, the safety and tolerability of this combination, and the proportion of patient who would have IC failure, and the time from tissue collection to to the molecular subgroup determination. It's what's the time frame look like, and there is a interim feasibility analysis at the end of stage 1. And we want to look at the turnaround time of these biomarker results. And we want to collect tissue from blood and tumor from these patients for future correlative studies. Next slide, please. The biomarkers used in this study are tumor mutational burden and nanostring TIS score. There is a cutoff of 10 mutations per megabase for TMB, and GEP, the nanostring score cutoff is 6. Based on these cutoff, we'll separate the patient into 4 different biological groups, and we'll recruit equal numbers of melanoma patients and head and neck squamous cell carcinoma patients into each of these groups. Next slide, please. The eligibility criteria is pretty standard. We will take patients with advanced melanoma. So uveal melanoma is not included here, but every other subtype of melanoma, and head and neck squamous cell carcinoma who have already progressed on anti-PD-1, and who have adequate tumor tissue to be submitted or to be biopsied. Everything else look pretty standard for immunotherapy trials. Next slide, please. So there are some, because cabozantinib is an oral drug, and it's not a drug that's typically is not approved for neither melanoma or head and neck squamous cell carcinoma. So we have additional language to for eligibility of for cabozantinib for patient using cabozantinib, and patient cannot have a history of hemoptysis or tumor bleeding within the past 90 days. There is some restriction to their prior radiation to the head and neck region, and they cannot have uncontrolled blood pressure. Next slide, please. So this is the, how the drugs are taken. So again, cabozantinib is an oral drug. So the patient was started with 40 milligram per day. If they cannot tolerate, then there are two dose levels that they can, the provider can reduce the cabozantinib to two dose levels. Nivolumab is given for 80 milligram IV every 4 weeks. So these are all pretty standard dosing. Next slide, please. So there are some adverse events that are more specific to cabozantinib, and some are more specific to nivolumab. And there can be some overlapping toxicity. For example, hepatitis, pancreatitis, diarrhea can be associated with both drugs. So there are very detailed instructions how to deal with these toxicities in the protocol. Next slide, please. So for the sake of time, I'm not going to go through these details in instructions. So these slides are meant to help the site staff to deal with these situations when they treat the patients. There are instructions for dose modifications. Next slide is for criteria to remove the patient from the treatment. And there are some instructions for registration, data submission, and also return of the results. So slide number 22 is the study calendar. So it's, you can see this is a pretty standard phase 2 trial. Patient only need to come in when they get the treatment, when they get the IV infusion. These are very standard procedure. There's nothing unusual about it, or there's no extra steps that need to be done. The only part that separates this trial from others is the tissue submission, both at baseline for eligibility and throughout the trial. So that, therefore, there are the next 2 slides. It's very detailed instruction how to manage these tissue collection and how to submit the tissue to the central protocol. So slide number 25 is the statistical considerations. Because this is a structure that has not been done before, and we're putting it together from scratch. So first of all, we want to ensure patient safety and make sure there's no futile treatment to the patient. So there are built-in feasibility monitoring, continuous feasibility monitoring, and interim futility and feasibility analysis throughout the trial. Even in step 2, there is interim futility analysis. And of course, there is a final analysis after all patients are recruited. So the next 2 slides is about operation, the funding and the resources for patients. So slide 28 is the study team for IMATCH. So I want to thank everyone on this slide from NCI side, from a SWOG side, who have worked really hard in the past several years to put this platform together, to put the pilot trial together, leading to the activation of the pilot last Friday. So the next slide is the contact information during the trial for questions. The last slide is really, I want to invite everybody to join the study team. It's a new way we're exploring. So we're not sure whether it's the right way yet, but we are hoping that you're interested in joining the effort to help find the best way to develop precision immuno-oncology treatment for our patients. Thank you so much. And both Katie and I are open for questions.
Thank you very much, Siwen, for that presentation. We're happy to take questions. So if anybody has any questions about this IMATCH pilot study, please, in the room, you can go to the microphones, or if you have a question, you can type it in the chat, and I will relay it. I'd really, while we're waiting to see if there are any questions, I'd really like to echo how it's been an amazing team effort and incredible to see the whole group work together. It really definitely takes a village to develop a trial like this. So I'd like to thank everybody involved. Siwen, I don't think there are any questions.
Okay, so if there is no further questions, I want to introduce our next speaker, Dr.
Oh, wait a second. A question just appeared.
Okay.
So I'm going to ask it then. It's in the chat. It said would it be valuable to use advanced renal cell carcinoma patients stratified similarly to act as a quasi-control group in the trial?
Yeah, that's a good thought. So at the beginning of the, when we put the concept together, there are a lot of thoughts put into which patient group we should recruit, right? Initially, we were thinking about recruit everybody, regardless what histology that is, because our hypothesis really, this mechanism, the combination, the synergy between these two drugs should not be restricted by tumor types, right? But it will be very hard if we do that. It will be very hard to get a conclusion in the end. So the biomarkers we're using, we're hoping, because we're recruiting equal number of patients in each biological groups, we're hoping that we can get tumor types or recruit tumor types that has equal number of distribution of these patients. So we don't end up recruiting, 3 of the 4 quadrants and then, have a hard time recruiting the last one, right? So there's very limited data to guide us on that, but there's some publication showing the distribution using these 2 biomarkers. What did the distribution look like in each disease group? And melanoma and head and neck squamous cell carcinoma is among those that has, relatively equal distribution of these patients, at least for IO naive patients. So that's why we chose these 2 histologies to move forward with.
Thank you, Siwen. So I'm going to turn it back to you to introduce our next speaker.
So our next speaker is Dr. Shirley Liu. So she is the co-founder and CEO of GV20 Therapeutics. And Dr. Liu she has a very impressive resume. She received her PhD in biomedical informatics and computer science from Stanford University. And she has been a professor of biostats and computational biology at Dana-Farber Cancer Institute and Harvard University. She was the director of the Center for Functional Cancer Epigenetics at the Dana-Farber and associate member of the Broad Institute at MIT and Harvard. So her research focused on algorithm development and data integration modeling for translational cancer research. Her group developed widely used algorithms for modeling transcriptional and epigenetic gene regulation, analyzing CRISPR screens, and tumor immunity. She contributed to the discovery of cancer drug response biomarkers, drug resistant mechanisms, and effective combination therapies. She has published over 250 peer-reviewed papers and have a H index of 106. So because of that, she received numerous awards. And on top of all of that, she has been a great mentor. She has mentored 25 trainees to independent faculty position. So Dr. Liu is going to talk about computational modeling of response to immune checkpoint inhibitors today. Thank you, Dr. Liu.
Great. Thank you, Siwen, for the introduction. Let me share my slides. Can you guys see the slides okay?
Yes.
All right, great. Yeah, so as Suan mentioned that I have been on the Harvard faculty for almost 20 years, and most of today's talk is going to be about my work at Dana-Farber, although I just recently joined GV20 Therapeutics as the full-time CEO. So we know that immune checkpoint inhibitors has brought paradigm shifts to cancer treatment, although only a minority of cancer patients can really benefit from checkpoint inhibitors. And so we are very interested in two major questions. The first is how to find the best indications or patients to use current checkpoint inhibitors to benefit. And another question is what kind of strategy can we use to improve the current ICI response? And so we are kind of previously in my lab, we were 70-80% computational, 20-30% experimental. And so today it's kind of a mixture of both, but more focused on the computational side. So we analyze many different data sets. One is looking at expression, other is to look at immune repertoires in terms of immunotherapy response. In terms of expression, we published this study back in 2018. We developed a computational expression signature called TIDE, which stands for tumor immune dysfunction and exclusion. And so we use this expression signature to predict the immunotherapy response from the pre-treatment tumor expression data from the tumors. And so basically, we evaluate the tumor for two types of expression signature. If the tumor is kind of immune hot with a lot of T-cell infiltration, then we have this T-cell dysfunctional expression signature. So this is a genome-wide expression signature. If the tumor is kind of a high T-cell infiltration tumor, we correlate the expression with this signature to get a score. When the score is high, it means that the T cells in the tumors are very dysfunctional. Therefore, even though it's an immune hot tumor, it's unlikely to respond to immunotherapy or checkpoint inhibitors. Whereas if it's an immune cold tumor with low T cell infiltration, we try to also generate an exclusion signature. This is again a genome-wide signature, which actually can help us evaluate whether the tumor has three types of immune suppressive cell types, which includes cancer-associated fibroblast, M2 macrophage, and also MD, myeloid-derived suppressor cell signature. So basically, if the tumor is immune cold and it has very high signature of either the fibroblast or M2 macrophage or MDSC, we believe that it won't really respond well to immunotherapy. Whereas even if it's the tumor is immune cold, it doesn't have those suppressive cell types in the tumor, it might still respond. And so with this signature, we also generated a website. And so this is called TIDE DFCI at harvard.edu. It's open to the public. And we realized most of the immunotherapy response biomarkers were generated using very limited cohorts. Therefore, we try to collect recently published immunotherapy expression profiles. Some of these are RNA-seq, some are nanostring. And so the genes that are covered are kind of different. And so by this time, we have collected, around 20 cohorts and close to 1,500 tumor profiles in the TIDE website. And so this has three different features. The first is a biomarker evaluation. This is mostly for translational scientists. So if somebody is proposing a new biomarker, instead of testing on like 2 to 3 cohorts, they can use all the TIDE cohorts to help evaluate. The gene could be a single gene, could be a signature, and also a signature would be kind of a list of genes. You can provide different weights, and we can use these weights to score the expression profile of each tumor. And so say a user can input this gene list with a different weight as the user kind of a custom gene set. We can use all the published cohorts to evaluate the predictive power in comparison with many other published biomarkers. The second function of TIDE is for response prediction. This is kind of for oncologists to look at the expression profile of a particular tumor and use multiple biomarkers to evaluate and see whether the patient is likely to respond or not. For example, this like one patient, if it has the tumor has high interferon signal or high MSI score based on the expression signature, high PD-L1, pretty decent CD8 T cells, and this is actually immune cold tumor, and not very strong dysfunctional score, and also very low T cell exclusion score, then we would believe this is going to be a responder. So we predicted it as a responder. And in reality, this is indeed a responder patient. And so you can see this is the overall TIDE prediction. If it's negative, it means that they don't have either T cell dysfunction or exclusion. Whereas if it's very positive, in this situation, you can see this tumor, it's an immune cold tumor. It has a very, very high T cell exclusion signature. And so TIDE has a very high score predicting this patient not to be a responder. And indeed, the patient is a non-responder. And so in some sense, we can use multiple biomarkers to evaluate the patient expression profiles and make predictions. And in a recent BMS Sage Bionetwork Dream Challenge, they asked different computational groups to make predictions of their Checkpoint 26 trial. And actually, the TIDE team was among the top performers of prediction. The third feature of TIDE is for regulator prioritization. This is most for cancer biologists. We want to use the public, immunotherapy trials and also the CRISPR screens and to see whether there are genes that are important as a putative target, which can be targeted to improve immunotherapy response. For example, currently, if we just run this, given a list of genes, we can see this. Actually, when we originally published the study, we were wondering whether glycosylation would be important, and we input all the glycosylation genes that are important, like known to be important for unglycan, and this MEN2A1 gene kind of actually show up really high, and you can see this gene is actually pretty high in a T cell dysfunctional score, and in many of the CRISPR screens, immune-related CRISPR screens, when we knock out this gene in the cancer cells, the immune cells can kill the cancer cells better. And also in immunotherapy trials, so this block is immunotherapy trial, we see that in some trials that the high expression of this gene is related to the worst response, especially in this case, a PD-L1 trial. And so we know that altered glycosylation is involved in immune suppressive signaling. And using this TIDE algorithm, when we look at all glycan pathway genes, this was actually the top #1 gene. And so we knock out this gene and look at the glycosylation patterns in the cancer cell, and we notice that overall, the complex glycan pattern in the cancer cells are decreased. And so we tested this gene knockout in the syngeneic tumor models. And we saw indeed that this gene knockout makes the tumor more sensitive to T cell mediated tumor killing. And so when we use a knockout gene expression signature from the cancer cell, so in the cancer cell, we knock out this gene, we look at the RNA expression profile to generate an expression signature, and we use this expression signature to look at association with immunotherapy response. We see a pretty robust separation of response for a PD-L1 trial from Genentech. And so we decided to test this in vivo because MEN2A1 has an inhibitor, that's a small molecule inhibitor. And unfortunately, as a single agent, it didn't actually pass clinical trial. So we are only testing this in combination with PD-L1. These are in two different syngeneic models. One is the B16 melanoma model. The other is the LLC non-small cell lung cancer model. And so you can see here, when we use either PD-L1 or the Swansonite small molecule to inhibit MEN2A1 alone, it doesn't have very, very strong effect. In fact, it's not too different from the control sample. However, when we combine the two drugs together, we really see a tumor growth control. In addition, when we grow the tumor for longer time, we can see also a difference in the mouse survival. And so we see that this combination actually can increase interferon gamma, TNF alpha without having more toxicity than the anti-PD-L1 alone. In fact, when we when we use this drug combination, we see that the tumor have overall higher immune cell infiltration and increased antigen presentation in the tumors. And so basically from the first part, we're trying to use expression to generate signatures of immune checkpoint inhibitor response. And we evaluated mostly a T cell dysfunctional signature in the immune hot tumors and T cell exclusion signatures in the immune cold tumors. And through this TIDE website, integrating many public data sets, we also identified a potential driver of immunotherapy response or resistance, which is MAN2A1, which is important for tumor glycosylation. And we hypothesize that when this gene is very high, the PD-L1 pattern or protein level on the cell surface might also be glycosylated, which makes the PD-L1 antibody no longer effective in binding to the PD-L1. Whereas when we use the swansonide to inhibit MAN2A1, the PD-L1 can actually present in the cell surface as its the form that antibody can recognize, and that will actually give us a more responsive tumor to the PD-L1 antibody. The next part, we also look at the expression profiles from the mouse tumors. So these are from a Syngeneic tumor database. Basically, there are mouse cancer cell lines which can grow in wild-type mouse tumors because they have the same genetic background. And so they have been extensively utilized in immune checkpoint inhibitor research studies. And so in this particular database that we call TISMO, we collected many different Syngeneic tumor models and also their expression profiles for both in vitro and in vivo. Most of the in vitro data are Syngeneic cancer cell lines treated with cytokine to measure gene expression differences. Whereas for the in vivo, these are tumors grown in the mice and treated with various checkpoint inhibitors. And the tumors were measured both in the isotype control as well as the immunotherapy treated. And for many of the in vivo data, we also check the original paper because sometimes even if it's the same model, they might, under the same treatment, there might be better responding tumors and worse responding tumors. And so we can collect that information. Because these mouse tumor data are much more better controlled, or the genetic background is the same, the cancer cell line is the same, we feel that if the mice are actually responding differently to checkpoint inhibitor, we could potentially really tease out the difference. Whereas with human tumors, the differences are the tumors are so heterogeneous, it's much harder to actually learn the useful signatures. And so with the TISMO, the collected expression profiles, we can allow users to explore a number of features. For in vitro, for example, these are, again, cancer cells, mouse cancer cells treated with different cytokines, and we can look at whether they have gene expression or pathway that are changing upon different cytokine treatment. Whereas for the in vivo data, we can have different features to look at whether a particular drug or immunotherapy treatment on these different Syngeneic models will give you, if you're interested in a particular gene or a particular pathway, or if you're interested in the immune infiltration level estimate, we can show from this website whether there is a difference between the responders or the non-responders, whether there's a difference based on the different treatment, or whether there's a difference based on the different cancer cell line models. And so with this model, we actually at this database collected 761 Syngeneic tumor models. And so this is a pretty big matrix of expression. The subject are the mouse tumors, and the genes are like 20,000 genes. And then we also systematically annotated their phenotypes. What's the cancer type, what's the cancer cell line, what is the perturbation? What is the mouse background or mouse strain, and what kind of immunotherapy were they treated with? And also for the response label, we know whether it's this particular cancer cell line or treated in the tumor model, whether it's a responder or non-responder. There are so many variables, but we saw actually, because they're only kind of because many of the mice are the same strain, same mouse, a lot of things are kind of related. Therefore, we try to kind of do dimension reduction to see whether there are features that are highly correlated, and see whether we can reduce the factors. Instead of having 20,000 genes, so many different phenotypes, we can kind of reduce this into kind of an expression signature, and also that is correlated with the response and also the phenotype. And so using this approach, we actually can derive expression signatures, and using this across many of the tied collected immunotherapy cohort, we were kind of happily surprised that the signatures we derived from very limited mouse, 600, 700 mouse models to predict immunotherapy response can actually separate many of the human tumor cohorts. Some of them didn't have enough patients. We don't have enough power, but for many cohorts, we actually have good predictive power of tumor response. In addition, when we look at the expression signatures and in terms of the prediction, we also saw that it can predict the cancer types that are more likely to respond to checkpoint inhibitors or PD-1 treatment. And so on the right side, we see melanoma, we see lymphoma, we see cervical cancer, head and neck, and on the left side, we see glioblastoma and also liver cancer. And this seemed to actually correspond quite well with the current response rate of the different immunotherapy studies. And also to our surprise, you can see here triple negative breast cancer, even with our expression signature, we could see that it will benefit more from ER positive breast cancer cases directly from the expression signature. And so we try to see, what are the signatures that are kind of interesting. For example, with melanoma, there is one signature which is correlated with better response. And you can see, not surprisingly, we see very, very strong CD8 T cell infiltration, TH1, TH2, TH17. This is also kind of CD4 related T cell signature, cell adhesion molecules, and also antigen presentation, immune regulatory interactions. So this is actually a good signature. And we want to also see whether there are gene signatures that are associated with worse outcome. And so you can see here, as another group of signature, we can actually have the genes ranked by the signature weight. And things like SOX-A, a collagen, like when we do pathway analysis, we see that signatures that are related to the ECM, extracellular matrix, to the Wnt signaling pathway, and the collagen are related to liver cancer and also a very poor immunotherapy response. Another one, which is in GBM, glioblastoma, this is a signature that is actually depleted in GBM, which is potentially related to a lack of immunotherapy response. And you can see here that in GBM, there's very low level of myeloid cell differentiation, low level of inflammatory response, and also lower level of these two genes, which are E3 related protein degradation pathway genes. And so we are hoping that, this not only provides this, like the mouse tumor model modeling, not only can tell us something about the immunotherapy response, get the signatures, tell us the indications where cancer patients are likely to respond to immunotherapy, but also give us some genes which potentially can be novel targets for improving immunotherapy response. And the third part we try to model is modeling based on cancer cell lines. And so we know that MHC class 1 is very, very important for the CD8 T cells in the tumor to recognize mutations in the tumor. However, class 2 presentation is also important, and normally they are being class 2 expression is very high on professional antigen presenting cells such as dendritic cell, macrophage, and B cells. But recently, it's also been reported that cancer cells, well, actually, once the class 2 presentation is high, it's presenting mutations, it can actually stimulate CD4 T cells. But recently, there are also published studies showing that cancer cells can also upregulate their MHC class 2, which can directly activate CD4 T cells. But then this whole MHC class 2 expression in cancer cells is kind of understudied. So we looked at 2 different big cohorts of cancer cell lines. And the first one is the CCLE cell line from the Broad Institute, where the expression of the over 1,300 cancer cell lines have been profiled. And if you look at the cancer cell line that have high class 2 presentation, the top is, lymphoma, myeloid, leukemia. So immune cells indeed have high class 2, but among the relatively cancer, like a solid tumor cancer cell line, skin or melanoma has very high class 2 expression. Another study was published by Genentech, where they have over 600 cancer cell lines. Again, after lymphoma, the next one with high class 2 expression is skin or melanoma related cell lines, suggesting that melanoma cancer cell itself can present antigens through class 2. And in a recent study, Checkmate 64 trial, this is in collaboration with the Dana-Farber Cancer Institute, it's been reported through the IHC staining that if the tumors have high level of MHC class 2 protein expression on the cancer cells, they actually would respond much better to the PD-1 drug from BMS. And so with that, we kind of also want to see whether this can be validated in a mouse model. So we grow the cancer cell, B16 cell, and then after that, this is a melanoma cancer cell line. We can sort the B16 cell by their cell surface protein expression of MHC class 2. And when we grow these B16 cancer cells into the mouse in the syngeneic tumor model, we can indeed see that when the MHC class 2 level is low, anti-PD-1 has very, very little effect. Whereas when the MHC class 2 is high, actually anti-PD-1 has an effect. So the B16 tumors are known to have an MHC class 1 defect. So it seems like the anti-PD-1 effect is very, very minor because of class 1 MHC presentation. Instead, the PD-1 might have an effect on the CD4 T cells. So when the cancer cells are presenting their mutations through MHC class 2, it might actually stimulate a CD4 T cell activation, which then can create T cell dysfunction. So having the anti-PD-1 treatment, it seems that the class 2 presentation on these B16 cells are really, really important. And so we are interested in asking what pathways are related to MHC class 2 expression on the cancer cells. This, again, we utilize a lot of public data on tumors and also cancer cell lines, and to ask what gene expression is correlated with the cancer cell intrinsic class 2 expression. And from the pathway analysis, we saw actually there's a very, very strong pathway, both, by the way, from the RNA expression data and the proteomic data, because the CCLE cancer cell lines also did a proteomic analysis of the different cancer cell lines. And both the RNA expression and the proteomic data point to the HEPO signaling pathway to be associated, especially there are some transcription factors, like also top genes like NF2 and RUNX1 to be kind of related to the MHC class 2, both RNA and protein level. And so in order to validate this, we use the CRISPR screens to knock out the genes that are really, really high, NF2, and also LATS1, LATS2, RUNX1. So these are all related to the HEPO pathway. So we look at the HEPO pathway. These genes are not only highly correlated, they seem to be important regulators. And indeed, we found, NF2, this AMOD-L2, and also LATS2, RUNX, and CBFB1. When we knock out these genes, they significantly decrease the MHC class 2 level. And also there's, these are the upstream factors. The downstream effector is a transcription factor called TEAD. And so the TEADs directly regulate MHC class 2 expression. We also try to look at published data. Indeed, when there was a separate study, when they knock down the TEAD, this gene actually suppressed the MHC class 2, and so it's a downstream effect. And we can see that when the TEAD transcription factor is knocked down, it can increase the MHC class 2 level. And so we believe, because the skin is facing outside and there are a lot of rigidity in the skin to form this structure, HEPO is kind of a pathway. When the cells are under pressure, when they're kind of squeezed, they get activated. So it could be that because the melanoma is facing outside, skin, there are a lot of kind of physical pressure involved. And so the HEPO pathway is activated to protect the melanomas from the outside physical pressure. But this in turn makes the cancer cell overexpress the MHC class 2, and that actually made the tumors more vulnerable to a PD-1 drug treatment. And so this is kind of our effort by looking at human tumors, mouse tumors, and cancer cell lines to model cancer cell response to immunotherapy, and also understand the potential mechanism of these response. The next, in a very short time, I want to also quickly kind of show you when we use immune repertoire modeling to understand immunotherapy response. So the first study is a collaboration with Margaret Shipp's lab. And so their lab actually discovered that the classical Hodgkin's lymphoma has a universal amplification of PD-L1 and PD-L2. And as a result, Hodgkin's lymphoma patients, when they are treated with anti-PD-1, they have the highest response rate of all the cancer types, with over 80% response. Of course, the remaining questions is still why only 80% and not every patient with PD-L1, PD-L2 amplification. And so in this particular study, the Shipp lab looked at the patient blood sample to look at a T-cell receptor diversity, like they did TCR sequencing from the patients. And so in here, they look at actually a number of patient groups. The first is healthy donors. And we look at the overall T-cell diversity, you can see that they actually have a pretty high T-cell diversity level. Whereas the newly diagnosed patient, they have much, much lower T-cell diversity. We know that in general, as people age, our overall T-cell diversity decrease, and also male decrease faster than female, which potentially explain why male are more likely to get cancer, because when they are older, they don't have diverse enough T cells to recognize tumor antigens. Interestingly, Hodgkin's lymphoma usually have a pretty early onset. Many patients are very young. They're like in their 20, 30-year-old, and yet their T cell diversity is actually quite low. Their T cells are like a very old person. And so maybe when there are mutations, their T cells don't really get to recognize that. And whereas if we look at the relapsed refractory patient, these are patients who have gone through 4 or 5 lines of treatment before PD-1 treatment. You can see that their T-cell diversity is even lower. Interestingly, when we look at the patient who were treated with immunotherapy, the complete responders, their T-cell diversity actually are pretty good. They're kind of more like the newly diagnosed patient, whereas the partial response or the progressive disease patient have much, much lower T-cell diversity, suggesting that having higher T-cell diversity is important for immunotherapy response, as at least in the blood sample, this seemed to be the case. And this is actually true for both CD4 and CD8 T cell. Like in this study, we did PBMC TCR sequencing for both the overall T cells, but also for CD4 separately or CD8 separately. In addition to profiling the TCR before treatment, we also profile the TCR after the treatment. So C1 is kind of the first cycle right after the first treatment, but we also look at cycle 4, day one. And so this is kind of the on treatment. We see, actually the overall T cell diversity increase a lot upon treatment. But when we look at, whether this is CD8 difference or CD4 difference, when we look at the overall T cells, we see an increase. When we look at just the CD4 T cell, we also see an increase. But when we look at the CD8 T cells, there is actually not too big a difference. In fact, when we look at the patient who are complete responders, they are the ones with increased T cell diversity, especially on the CD4 T cell compartment. Whereas for the partial responders or the progressive disease, we don't actually see a very strong difference in their T cell diversity. So actually with the Hodgkin's lymphoma, the cancer cell is this Hodgkin-Reed-Sternberg cell. They have very frequent B2M mutations. So again, their class 1 antigen presentation is lost in this case. And so the cancer mutations are not able to be recognized by CD8 T cells. Instead, the Hodgkin's, the Reed-Sternberg cells can express MHC class 2. Therefore, they are seems like they're being recognized by the CD4 T cells. And that's why after immunotherapy treatment, the CD4 T cells in the blood seem to really get a chance to be activated and also increase their T cell diversity. So from this analysis, we see that in general, at least for Hodgkin's lymphoma, if the patient has a higher T cell diversity, it indicates that their overall immune health is probably in better shape. And also after immunotherapy treatment, if their diversity can increase in the blood, it also is an indication of a better immunotherapy response. Next, we also try to model the B cells in the tumors. In recent years, there has been a gradual realization that B cells in the tumors are very important. So in 2020, there are 3 co-published Nature papers to show that B cells and the tertiary lymphoid structures are very important and associated with immunotherapy response. So if they are just immature tertiary lymphoid structures, scattered T cell or B cell kind of zone, they seem to have a more immune inhibition effect, whereas the mature tertiary lymphoid structure, they seem to be associated with better immunotherapy response. And so we actually looked at some of the non-immunotherapy trials before. So this is just from published tumor RNA sequencing from AML. And these are actually not treated with immunotherapy. They're actually treated with chemotherapy. And so we looked at their B cells in general. And so these includes the AML from infants or children, like pediatric AML versus adult AML. So interestingly, we saw that there is actually one factor which can really distinguish patient outcome. So in the AML samples, there are actually antibodies in the blood. And if the antibodies have high IgA levels, so in infant, most of the healthy individuals, they don't really have IgA antibodies at all. They have mostly IgM and maybe a little bit of IgG antibody. They have very, very little level of IgA. However, with AML patient, this shift, actually happen much earlier. So normally when people age, their naive B cells are decreasing, so they have less IgM, they have more IgG, they have gradually more IgA antibody. But when you have AML, this shift happened much earlier. Infants start to have IgA1 antibodies, and high IgA1 is associated with worse patient survival. And in adults, this shift happened even stronger, and patients are starting to have the late IgA2 antibodies. And again, if the IgA2 level is really high, they have overall worse clinical outcome. We actually look at what are the factors that are potentially important. So previously in 2015, there is a Nature paper published by Michael Karin's group. They found that there are cytokines such as TGF-beta, which can induce the plasma cell to shift from a IgG kind of producing plasma cell to an IgA producing plasma cell. And this seemed to suppress the overall immune function in the tumor. And so we asked in those AML patients, do we really see this indeed, we see that even in the AML sample, not solid tumor, that TGF-beta level is correlated with the IgA2 level, which suggests that sometimes in the tumor microenvironment, if TGF-beta level is high, this actually can be cytokines secreted by the cancer cells. It can then induce the B cells to go from an IgG antibody to an IgA antibody, especially the IgA2 type, which can really suppress immune function. And so indeed, in the adult AML samples, we can see when the IgA2 level is high, FOXP3 level is much higher, suggesting that in those IgA tumors, there are also stronger Treg infiltration, which is more immune suppressive. And this is not only in AML samples, but also in solid tumors. And we look at TCGA tumors, and there are 3 cancer types where the overall IgA levels in the tumors are worse, indicates a worse survival, whereas IgG3 and IgG1 like isotypes are actually associated with overall better patient survival. So actually in the patient tumors, we know that they are tertiary lymphoid structure. They are creating the B cells, having the clonal expansion to produce antibodies. And if the antibodies are made to produce IgG1, IgG3, these are the 2 isotypes that are more likely to have a complement, like a recruit complement, also ADCC to activate NK cells, and also ADCP to activate macrophage. These are the anti-tumor macrophages. And so you can see here, if we look at the overall IgG3, IgG1 level over the overall like antibody signal, heavy chain level, you can see here when the patient have higher, IgG antibody in ovarian cancer, melanoma, and bladder cancer, they would have better survival. And so with the tight collected immunotherapy trials, we also look at their Ig or kind of expression, and also the T cell diversity, B cell diversity, and also isotypes. So here we don't have as many trials as we have in TIDE, because many of the TIDE signatures are also from nanostring, and for those, we don't really know their B cell or T cell signals. But overall, if we look at all these different trials, there are pre-treatment samples. There are also some cohorts with on-treatment sample. And we are looking at what's the difference between the responders and the non-responders. And, do we see coherent differences? Overall, we see that if the tumor have overall higher B cell abundance, they will be more likely to be a responder. If they have overall higher like a richness of antibodies in the tumor, they also will likely to be like a responder. And also if there are overall higher clonal expansions in the B cells, they also are corresponding. So if the antibody have a chance to do clonal expansion, they are more likely to be a responder. And if they also produce more of the IgG1, IgG3 isotypes, they are likely also to be a good responder. And also very often the high clonal expansion is associated with low diversity, because certain clones that potentially recognize the tumor antigen are getting a chance to be clonal expanded. And so they have lower diversity and higher clonal expansion. And so in a recent immunity paper that was published, where they used spatial transcriptomics to look at the tertiary lymphoid structures in the tumor, we actually wrote a highlight of this study. It was a very interesting study here. They found that there are tertiary lymphoid structures in the tumor, and you can see the T cell zone and the B cell zone. And so in the B cell zone, the B cells are going through clonal expansion and somatic hypermutation to evolve antibodies. And then those B cells that are like maturing into the plasma cells then migrate through the fibroblasts into the tumor niche. And interestingly, they found that if the B cells or the plasma cells here are producing IgG antibodies, then the adjacent cancer cells are more likely apoptotic, meaning that those antibodies are likely to be anti-tumor. Interestingly, if it's a a B cell that's producing the IgA antibody, then they are kind of scattered, and the nearby cancer cell are no longer being killed by the plasma cell or the antibody. So it would suggest that within our tumors that are tertiary lymphoid structures creating antibodies, and these could create antibodies that are either pro-tumor or anti-tumor, and the IgG antibodies are kind of anti-tumor, but the cancer cells can secrete cytokines such as TGF-beta, which kind of induce the B cell to shift from IgG plasma cell to an IgA plasma cell, and then the antibody will lose their tumor killing effect. And so, so to summarize that overall, the overall B cell level, tertiary lymphoid structure, and also the level of IgG that's produced by the B cells in the tumor are associated with better immunotherapy response. I think that's kind of our current effort. I think there's still a lot of work that needs to be done to better understand immunotherapy response. And so most of the work is done at the Dana-Farber, and these are all the lab members that are involved in the various studies and with the help from other lab members. And also I want to thank our collaborators, especially Miles Brown, Steve Hody, Kai-Wookie Fennick, and Margaret Ship from the Farber. So I think I will just stop here for questions.
Thank you so much, Shirley. I don't know whether there's any question from the site, but let me start with a question, Shirley. I have been so impressed with your effort to harmonize the public data, right? And take good use of already published data, and always publish your efforts, make public available database. So my question is, as someone who don't completely understand how you harmonize those data, right? But I often hear from, the biostatistician or bioinformatician I work with, if the data is from different platforms, right? If they are done, especially for human data, right? It's if there are the original tissue is from different kind of tissue, whether it's FIP or it's a fresh frozen tissue. And also, the platform they use to sequence the tissue can make huge difference, in terms of the final result. And it's very hard to compare, across different platforms. So how do you encounter those problems in your efforts? And what would be your thoughts on harmonizing those data?
So yeah, very, very good question. Indeed, depending on the cancer type, depending on the platform, depending on whether it's fresh tissue or FIP, the results are really, really different. Our current effort is just to collect and process the data. We are not trying to harmonize between different studies. We do use the same signature in different cohorts just to see whether if we would believe that if the signature is robust across as many cohorts as possible, then it's a real one. But we are not trying to kind of normalize between different cohorts because it's really, really hard.
Okay, yeah. So when you showed at the beginning, there are probably 20, 30 different studies and that you were looking into, you were analyzing these signatures in each of them.
Yes. Yeah, we were not trying to so each of the study you can see here, each line is one cohort, and you can see whether that signature has predictive power in that cohort or another cohort. We're not trying to combine the different cohorts in here.
Yeah, thank you. That clarifies things. So another question is about, your interesting observation about T-cell receptor diversity and, Hodgkin lymphoma is all young patients and, the diversity is low. Do you think that's a Hodgkin lymphoma phenomena, or do you think it's, just those patients who have lower TCR diversity tend to get cancer? It might be a cross-cancer thing.
Yeah, so previously we looked at the TCR diversity of 600 normal individuals, and you can really see a very good separation with age. people's T cell diversity decrease, and also male decrease faster than females. So I was like, wow, we should probably do our TCR sequencing every 5-10 years to make sure we are on the right trajectory. And I think if their T cell diversity is much, much lower, then mutations in the cancer cells will become invisible to your immune system, and they will, get a chance to continue to grow. And I think, of course, in other cancer types, it's also possible that T cell diversity is an indication of age. And we know that, young people have better immune system. They probably would respond better to immunotherapy. But yeah, I do think that T cell diversity in the blood is a pretty interesting measure of this person's overall immune health.
Yeah, that's very interesting. So Katie, is there any question from the site?
There is a question from the audience.
Yes, hi, this is Rafi Nakash. Thank you, Dr. Liu and Dr. Liskowin. So a couple of questions. One, Dr. Liu, is the slide that you showed with the TIDE algorithm predicting patient responses. There were, I think, 7 patients that you had some false positives. So what was the reason? Did you happen to dive in deeper why the algorithm didn't work there?
I mean, so far there's never any biomarker that can robustly predict everything. For example, I mean, you can imagine, let's just look at this particular patient. They have very low interferon score, they have a high MSI. And so even though the PD-L1 CD8 level are kind of low, it's still a response. I guess this might be a colon cancer case, or I guess because the patient has still an overall higher immune infiltration, and I guess the suppressive signals are not that strong. In here we can see things as strong as like a 2.9. Here the exclusion score is normalized. And so, we might have a little bit of a suppressive macrophage here, but it's not as strong. So maybe the patient still have a chance to respond, even though it seemed to be an immune cold tumor. But frankly, I think in general, we are not seeing enough of these contradictory cases to really learn the rules. Because the human tumors are just so heterogeneous. That's why we thought maybe mouse tumors would be better, because they all initially, the soil and seed, the mouse and the tumor, we are better controlled. Maybe we can learn some of the subtle differences better.
Sure, thank you. So second question is related to TCRs, and there's 2 questions there. One is, and I guess this is partly a question for Dr. Liskowin and yourself also. So with the recent neoadjuvant therapies that have come up in lung or melanoma or many other tumors, some of the hypothesis is around that lymph nodes are preserved in the neoadjuvant setting, so there's more antigen presentation. Is there any data to suggest that there's a higher TCR diversity in patients post-resection of tumor and lymphadenectomy that could be a reason why neoadjuvant therapies have such a significant response, possibly also related to the more neoantigens that are present? And the second part of the question is, there's obvious bioinformatic pipelines trying to predict MHC peptide binding affinities. Is there any, from your experience, Dr. Liu, is there any bioinformatic pipelines assessing which neoantigens can lead to a higher TCR diversity? And I'm not specifically referring to like an interferon gamma ELISA assay, but more like a TCR assessment if certain neoantigens can actually expand that diversity in some way.
Yeah, maybe the second question, frankly, I don't know the answer. I don't think people have really matched the neoantigen to the TCR diversity. But in terms of Hodgkin's lymphoma, I have a suspicion that in the tumors is not just neoantigen, there are also autoantigens. In fact, in this particular study, we saw some interesting trend, but reviewers feel like we didn't have enough data to make a conclusive result. But actually, some of the CD4 T cells, we could match their CDR to a known antigen. And turns out they are autoantigens. They're not new antigens, which is kind of a surprise. But maybe that is indeed the case. Yeah, and so also we feel that if the patient blood has higher T cell diversity, it's a good thing. It means that they have many different T cells to recognize any kind of mutations. But in the tumor, it seems that like a clonal expanded T cells in the tumor are more indicative of better response, because it means that the T cell has already detected that antigen, whether it's a new antigen or a autoantigen.
I guess the first part of the question was more related to lymph nodes and TCR diversity. Not sure what your thoughts are over there.
Yeah. Yeah, I mean, I think that there are reports showing that the draining lymph nodes have T cells and B cells that get preserved, or it's a reservoir for infiltrating into the tumor. I haven't seen a lot of TCR sequencing of the draining lymph nodes being published, so I don't know whether there are any known associations. I don't know whether Siwen has seen some recent studies.
No, I have not. I think that would be a very difficult study to do, especially, in human, right? Yeah, I'm hoping maybe 1801, correlated study can help us to understand some of these. Raphael, it's a very good question you asked. So thank you so much, Shirley, for presenting today. And it's very relevant to our topic today. And I will elect Katie to introduce our next speaker, Dr. Sean Bendel from Stanford University.
Yes, thank you very much, Siwen, and thank you very much, Dr. Liu, for that great presentation. I know I have a lot of follow-up questions that hopefully I'll be able to ask you sometime.
Sure. Thank you.
So, our next speaker is Dr. Sean Bendel. He's an associate professor in pathology at Stanford University. Dr. Bendel specializes in the development of single-cell proteomic tools for the investigation of human systems, including pioneering single-cell cytoff mass cytometry and multiplexed ion beam imaging. Sean's work provides unparalleled granularity of understanding in multiple facets of human stem cell systems, hematopoiesis, and immunology. And his lab continues to unravel both healthy and dysfunctional human hematopoietic immune cell biology, as well as the cellular landscape of cognitive decline in the human brain. He's received various awards, including being recognized as Damon Runyon's Breakthrough Scientist and an NIH New Innovator Award. So thank you very much for speaking today.
Thank you. Is that good? Is there a clicker?
There is a clicker.
Does it work?
It does work. And would it be possible to show the slides, please?
I can see it. Well, yeah. Oh, for the Zoom, yes.
Yeah, for the Zoom. Full screen.
So yeah, thank you for the intro. I guess Jerry.
A second, sorry. I think
All right.
the Zoom isn't showing the slides properly, I think. Or at least not for me.
It is showing.
You just see the main screen?
Yeah.
Okay,
Yeah,
great. Okay.
left. Yeah,
I guess
perfect.
it's just me.
Well, thanks for the intro, and thanks for inviting me to share, to get on my soapbox and tell you a little bit about some experience we've had with using single cell technologies for immune monitoring in various immunotherapy trials. So I've got a couple stories to tell you about today. And quick disclosure, I'm still a scientific consultant, work with IonPath that makes some of the multiplex imaging technologies in my presentation today, but all the work's come out of my lab. So this is actually a kind of a nice segue from, so Shirley and I actually have a connection in the past, and it's through this CMAC network, which many of you might be familiar with. This is came out of the Cancer Moonshot, and it's currently being renewed, and we are one of 4 analysis centers at Stanford. And the idea here is we're supposed to be working with trials like the IMATCH trial that you guys talked about today to help bring the best kind of immune monitoring and analysis tools so we can figure out interesting biomarkers and understand the same things that Shirley was talking about in the last presentation about how can we understand who's responding to therapy, why people are having adverse reactions, and better ways to kind of put patients in the right bin. And I'm the co-investigator with Holden Maker, who runs the Human Immune Monitoring Core at Stanford. But we have we work and collaborate with centers and share assays and samples between us, the Farber, Mount Sinai, and MD Anderson. And actually, prior to leaving Dana-Farber, Shirley was actually running the CIDC, which is the core that's catching and integrating and organizing all this data for us. And so, just to give you an idea, even though I'm a single cell zealot. My lab does a lot of single cell technology development, different ways to ask and answer questions about the immune system. I kind of wanted to bring up this list of assays here, which is what a lot of the CMACs have agreed we should be trying to pair up with the various clinical trials that we're working with. And the ones marked here with the star are actual kind of have single cell data in them. And I just want to point out that a lot of the time single cell technology isn't necessary to get lots of information. And in fact, a lot of the cool insights that Shirley showed in her last presentation were using bulk data analyses, where she does have some interesting ways to do single cell convolution and figuring out signatures from individual cells within the samples, but you don't always need to do single cells, because as this indicates here, particularly with some of the sequencing-based assays, they're quite expensive, and actually dealing with the samples in a way that's compatible with the downstream analysis can also be tricky. And I'm going to come back to that later. But what I'm going to focus on right now is some technologies that have largely come out of my lab that's being used and beyond Stanford in this network. And that's a combination of cytoph mass cytometry, a next generation single cell flow cytometry based assay, as well as multiplex ion beam imaging for doing tissue IHC. And both these rely on the use of metal-based reporters in the place of light reporters. So maybe you've heard of these before, maybe not, maybe someone even has one of these instruments in their lab. But the core of the technology really is, you think about your assay where you do immunochemistry or have some sort of probe, and normally you'd have an enzyme or a fluorophore on that probe. And instead, all we've done is we've come up with ways, or worked with people who've come up with ways to take the fluorophore or the light-based probe out of the assay and instead insert a non-biological elemental isotope. And we're just reading these things out on a mass spectrometer. But the idea is the data that comes out of these assays looks very much like the data in your conventional light-based assays. And so this is just a schematic of cytoph mass cytometry. But the take-home here is that when you're thinking about running this on multiple time points, dozens of patients, that you can use a platform like this, and hopefully I can convince you with a little follow-up here, that you can routinely get highly multiplex single cell analysis. It's a platform that allows you to measure millions and millions of single cells a day. It can be quantitative and robust, and that the important part is when trying to figure out how to do all of this, it only costs you a fraction of a penny per cell in your experiment, right? So this is why this particular assay is a tier 1 assay in the CMAC network, because it's something where, working within the NCI support and guidelines, we can offer this to every patient peripheral blood sample on every trial that we're working with. And just to kind of give you an idea of the type of assay we've put together to offer, this was a study that my lab put together, where what we wanted to do is we wanted to come up with a one-stop shop for immune monitoring for mass cytometry. And so Felix was the postdoc that led this, but again, this was in collaboration with a lot of CMAC centers, as well as the Parker Institute for Cancer Immunotherapy. And so the goal was to kind of demystify panelification of these cytometry-based workflows. If you guys have ever gone down to your flow cytometry center, talked to your favorite immunologist, everyone's got their favorite markers, their favorite ology, their favorite fluorophore instrument. There's all this mysticism around cytometry. And so what we wanted to try and do is cut through a lot of that and get down to what's a basic panel that we can recommend that leaves enough room for customization for your particular assay or interest or cell type, but covers enough of the immune system so you don't really have to think about or worry about capturing cells that you might not anticipate being there in a particular patient study. And so, again, all of this is published, it's online, but But the kind of take home here is using about 30 features, we can not only catch our immune phenotypes, but also cell activation and behavior, immune checkpoint markers, and things like important transcription factors that define regulatory states like Tregs, for instance. And so what does the data look like when you put all of this together in a tube? Well, you can go through and you can look at it in two-dimensional scatter plots, and you can find all your favorite immune cell populations. You can take this 30-dimensional data and you can apply something like a UMAP or a TISNI. You see a lot of these coming up in immune monitoring papers, but essentially this is your 30-dimensional data in a two-dimensional space where we can see the different islands or different cell populations and states. Or you can cluster them and analyze them and compare these clusters between your different studies. But I think the most important thing is to circle back to what the original goal of putting this together was, which is how broadly are we capturing the immune system without kind of coming in with any priors. And so the way we designed this panel and what we were able to show here is that essentially 99.7% of all immune cells in all of the patient samples we optimized this on had 4 positive identifying markers or more. And then we could break this down by individual cell population, but the point is that you're pretty much not going to miss an immune sample or an immune cell type in a cryopreserved PBMC sample if you're using an approach like this. And so this is kind of a custom mix that we put together, but what the CMAC is using is a small variant of this, and there's commercial resources to actually purchase this from the vendors that support this instrumentation. And so I think the other important thing is not just what fancy analysis can you do, but how routinely can you do it? This is a question that you should be asking when thinking about, incorporating a technology into a trial or an immune monitoring study on your patient samples that you have, painstakingly collected and organized from your different centers that are running things with you. And here you can see in the top left, we get quite good correlation between running technical replicates at different centers by mass cytometry, biological replicates and technical replicates cluster together, and we can even benchmark with an orthogonal flow cytometry technology and show that we get kind of 1-to-1 on the same sample. And it's not in this slide here, but we've also published a follow-up study with the different CMAC centers actually showing concordance between the different centers running the same assay on the same samples. When you have something like this, then what it enables you to do is then we can go at a biological problem without any prior assumptions about what might be going on here. So this was just an example of a local study we did at Stanford where we were working with the bone marrow transplantation group to try and understand immune changes associated with graft versus host disease. And I think if you're thinking about GVHD, oftentimes people think about myeloid cells, they certainly think about cytotoxic T cells, but lo and behold, what it turned out here is that when we ran this panel on a small collection of patients, the strongest signature actually came up with being with having naive CD4 cells and naive B cells being more associated with a protective state against GVHD and BMT. So the other aspect of this formula is going from a liquid sample like PBMCs that you might cryopreserve to a solid tissue sample, something like an archival FP tissue section. And so this has inspired the creation of adapting this assay to a solid phase format. And we've actually built instrumentation, and this is what the company IonPath now commercializes. But this is something called multiplex ion beam imaging. It's another type of fancy mass spectrometry, but the long story short is it's kind of like a digital microscope. That's how it feels like when students and researchers are using it in the lab. But essentially what you do is you take these non-biological elemental reporters that are attached to antibodies. We stain FFPE tissue sections. Those sections are loaded in the slide. And an ion beam rasters over that section with nanometer precision, and you get a 2D image that has the location of your different reporters on there. And again, we can stain 40 to 50 things at a time routinely. And what this allows us to do with this next-gen instrumentation versus one of our older kind of proof of concept of the assay is we can pretty routinely scan quite large areas of archival tissue and do what we think is close to a comprehensive immune cell immunophenotyping. And just to kind of throw in, again, a little validation data, because this is something I think, even though I think validation data it's a boring part to do, but you want to, before you, I guess the thing I always tell students in my lab is, before you can show me something's different, show me that you can find something is the same, right? So like running replicates and running controls and showing that you get the same data over and over, it's as boring as it sounds, is a really critical feature to being able to show that you're sensitive to find changes in these sorts of assays. And so this is just a study that we ran with the CMAC network, actually using a collection of carcinoma samples put together by the NCI, where we're looking at replicates of the different immunophenotypic markers that we quantified simultaneously. And both at the kind of tissue pixel level, but also at the cell type level, we can get quite routine reproducible data. And we've gone beyond this now, where we're actually not looking beyond just maybe as an assay itself, but just benchmarking multiplex IHC. So this is us working on the same sample types with the different CMAC centers, NCI, at Dana-Farber, Mount Sinai, and MD Anderson, just to show that when we actually run serial sections of the same archival samples, we can also enumerate, produce the same data. So this is an important piece of the puzzle if we're all going to be able to run these kind of immune monitoring efforts and do biomarker discovery across these networks. So now to kind of more fun stuff, what kind of new things can we do? Because I think it's one thing to kind of count and enumerate the immune cells, and you definitely want to make sure you're not missing things, but are surface markers good enough, right? We know ultimately the receptors on the surface of a cell, the things we use to phenotype and identify them, control how they interact with each other, how they talk to their neighborhood, and ultimately their function. But is there other features intrinsic to these cells that can tell us a little more about what they might be doing? And so this is another, Felix, also the now assistant professor who did the cytoimmune benchmarking work. He also had this idea that maybe we could start to, in an archival retrospective format, make some estimates about cell metabolic state in combination with cell identity and immunophenotype. And so the goal here really was, so I guess to take a step back, normally cell metabolism's measured or quantified by looking at cellular metabolites, right? There's assays out there called metabolic flux analysis, this Agilent Seahorse platform that does a colorimetric measure of glycolysis or oxidative phosphorylation, as well as more sophisticated formats. You actually have people by mass spectrometry measure and identify the different metabolites the cell's making or producing, and that gives us an estimate of overall how it's creating and using energy. That's really hard to do in an archival fixed sample or a crop preserved sample, because the cells have shut down, often the metabolites are gone. And so Felix got this idea that instead of looking at the metabolites themselves, we could look at the rate limiting enzymes and transporters in these metabolic pathways, right? Break out your first year med school biochemistry or Biochem 300 textbook and look at the key things in these pathways. And if we measured enough of them, or we measured the right ones, that they would be actually predictive to the overall metabolic state of these cells or tissues. And this is just an example of the kind of the rate limiting things that we boiled it down to. But to kind of make a long story short, what we tried to do is benchmark this approach in terms of single cell measurements against these gold standard assays. And what we were able to show is, at least in terms of glycolysis or oxidative phosphorylation, so it's kind of more your like TCA cycle, electron transport chain, for both of these main metabolic pathways where there's gold standard colorimetric assays, we were able to get one-to-one correlation with single cell approximations of these same things. So that was sort of fun. But the other cool thing, or important thing, when you think about this in terms of an application of biomarker discovery or immune monitoring, is that no matter what time point the cells sat for, or how long we collected, whether we let the cells sit in the fridge for 4 days over the weekend, these metabolic signals, because they weren't based on the metabolite themselves, but the wiring of the network, they seem to stay relatively stable. So we got the same answer on Monday, even if the sample was collected on Friday, for instance. The other thing that is important to note is when you deal with healthy normative cells from the same tissue between individuals, the metabolic network is quite conserved. And so it seemed like the metabolic network in a B cell versus CDC versus a basophil they were unique from one another, but conserved across individuals, right? So my B cells are metabolizing like your B cells and so on. And so that was another important feature. So when you kind of put all this together, we took a little bit of a dive into looking at cytotoxic T cells in tissue. And so we did an analysis where we looked at peripheral circulating T cells that were dissociated from lymph nodes, as well as some paired healthy and adjacent colon carcinoma T cells that we had looked at there. And a couple things came about when we zoomed right in on the T cell island here. And so what you can see here on the right is this is kind of a dimensionality reduction of all the metabolic usage of just the CD8 T cells. And when we go in there and dive in and kind of look at what are the different metabolic T cell states we see, a couple things pop out. So one, when you look at healthy tissue, so look at whether the peripheral blood there at the bottom on the left or healthy colon kind of on the left, you really do see quite consistent distribution of metabolic pathway usage across different healthy individuals. But when you move into the carcinoma sample, that's where you start to see more variability. And there's certain metabolic clusters of T cells that pop up uniquely in the tumor itself. Mainly this maroon one and this yellow one, cluster 3 and cluster 9. And we call both of those out, one, because they're unique, but two, because they're almost like polar opposites of one another. Cluster 9 is metabolically extremely active. It's undergoing both, it has both regulators of aerobic and anaerobic respiration upregulated, where cluster 3 is really quiet. It has some receptors on the surface for amino acid metabolism, but other than that, it's crickets. But the other reason why those two are interesting is because when we zoom back to kind of our immunophenotypic markers, things that we typically use to ask questions about the immune microenvironment, they actually look more similar, right? They both have PD-1 and CD39. They're both expressing what we think to be dysfunctional markers, though there is some heterogeneity in which ones are more proliferative by Ki67 and TCF1. And so not surprising that the kind of T stem cell factor or Ki67 was more upregulated in the more metabolically active cells. Surprise, surprise. So what does it look like when we actually go take a peek for those cells in situ in the tumor microenvironment? And so what we found is when we went back and looked at paired samples from these same resected tumors and actually looked at where these cells were, it might be not surprising to anyone here that they weren't necessarily in the same place. And so it turned out that the cells that were more metabolically active, and these are the ones in red here in the image, these ones were much more close in proximity to the actual tumor itself, versus the ones that were metabolically low, this kind of cluster, dysfunctional looking cluster 3. These cells were in the tissue, but they weren't engaged. They were quite far away from any actual tumor margin. And so I think, kind of putting this together, I think this shows you how you can go beyond your traditional immunophenotype and start to think about using other molecular features or proteomic features of cells as a means by which to kind of maybe deconvolve confusing or conflicting observations with, like, say, for instance, checkpoint expression, but also that there's other levels of cell activity or identity that's still available to be found beyond just, say, your CD4, CD8 sort of thing. And in the few minutes I have left, I wanted to kind of tell you one more story where I'm sort of a minor actor in this one, but this is kind of a recent immunotherapy and immune monitoring story that's come out of Stanford. And this is around kind of a soup to nuts approach with the CAR T cell therapies that have been going on there led by Crystal Makel and Dave Nicholas. And so this picture, I think, really does it justice in terms of, this is the off-site, which is actually on-site because, all the clinical staff can't actually get that far away from the clinic. The Center for Cell Therapy retreat that we had, I think it was last year. And it just gives you an idea of how many people are required both to manufacture these cell therapy products, to deliver them to patients in the clinic, and then the whole cabal of people required to actually run around and do sample and biomarker collection and analysis from these patient samples. And I'm going to give you a little my 2 cents commentary along the way about the different assays and how they were utilized. And so this particular study, this particular paper was focused around the AxiCell product. This is the Zuma-1 trial. So this is actually, we weren't making these particular CAR T cells. These were CAR T cells from Kite. And this is the anti-CD19. And the two kind of core questions that Zena Good, who was the lead author on this study, working with clinician Jay Spiegel, were trying to get at was, obviously, we know this therapy can work really well for lymphomas, but it only works well for about half the patients. So why only half, right? Is it something about the product, or in this case, is it something about how the product behaves in the patient? And then the other thing, of course, is there's issues around toxicity, particularly neurotoxicity. And so these were some of the kind of, I'd say, clinical metadata that they were putting up against all the biomarker data that we were trying to collect. And for this story, I kind of want to go from the beginning to the end, which is tell you the answer and then give you a little context about how we got there. It turns out one of the big signatures that popped out in all of the different omics we were applying to this was that there were these CAR Tregs that were showing up and expanding in the patient early on, day 7, day 21, that seemed to be both associated with negative clinical response. So basically not, the CAR failing to kill the tumor, but also seemed to also paradoxically, or maybe not, you'd actually expect this, seem to have a protective effect against the neurotoxicity aspect. And all of this seemed to be not related to the CAR cell expansion itself. And so the way this study was done, and the way we've been actually handling all cell therapy patients coming through, at least the ones on any studies coming through Stanford Hospital and clinics, is we have a SWAT team of nurses in the clinic that work with research staff on what we call the Center for Correlative Science that worked with CCT, and we collect blood samples at essentially pre-infusion, post-infusion, and then at the earliest time points to really understand how the patient tumors or the patient product is expanding. When we can, we obviously get archival biopsies of the patient tumor, as well as when we're manufacturing the product themselves, we're also banking the leukapheresis product as well as the expanded CAR cell product from those patients. And we're trying to kind of capture and preserve all of this in a way where we can apply as many assays that we can think of, even if we haven't thought of them ahead of time. But what I can tell you is that for some of the assays that we're running, particularly some of the single cell sequencing assays, these assays require patient samples to be run, or at least optimally run, fresh. And so these require a high degree of coordination, including having technicians to do sample prep, working on evenings and weekends, which, it actually is both a huge coordination effort and a huge economic cost. The other thing that it has enabled us to do, though, is besides doing kind of more sophisticated biomarker discovery retrospective assays, it's also allowed us to do a little bit of real-time monitoring, where we've come up with low-plex versions of some of the assays we're doing just by conventional cytometry, where we can actually report back within a day or two to the clinical team treating the patients levels of CAR in patient's periphery and other features of their immune system, so they get some ideas of what's going on. And particularly monitoring just overall peripheral CAR T cell retention has been, I think, helpful. It's a metric now that a lot of the oncology staff ask for. And so, like with the CMAC assays, the CCSU, one of the things we run on all our patient PBMC samples and our CAR T samples is this kind of core CyTOF panel that we put together. This one's been modified a little bit from the one I showed you at the beginning, where we've had a higher focus on T cell exhaustion and functional markers, as well as for each of the CAR trials, we try to, and in this case, have developed an anti-idiotype antibody. So this is an antibody that recognizes the CAR CD19 receptor, so we can actually tell a CAR T cell away from a native T cell. And so what we did is, after all the samples were actually collected and banked, so this is all PBMCs for this particular particular analysis, samples were thawed in batches, and we ran in kind of barcoded batch runs all of the CyTOF work using this panel. And then Zena and the computational team applied various clustering and machine learning approaches to this. And I'll tell you, you always see a way in a paper, or like what Shirley showed you in the last talk, something that looks really straightforward. But what I'll tell you, what really happens behind the scenes is we try a whole bunch of stuff, and usually something sticks, and we try to validate it orthogonally. But in this case, Zena came up with a few clusters from the CyTOF data of these peripheral blood samples from day 7 and day 21 that were predictive in helping us to differentiate the response of patients from people who had progressive disease. And the initial kind of clusters popped up with these definitions here. And there was kind of 2 positive clusters that were both CD4 and CD8 CAR T cells bearing CD57. This is kind of a cytotoxic cell marker, same molecule that NK cells use. And then there was a progressive disease associated marker. This was the Helios positive CD4 T cells. When we dug into these cells a little bit more, it turns out that these CD4 Helios cells were also FOXP3 positive. I don't show the data here, but it's in the paper. We actually did some follow-up assays where we actually showed that those cells have the potential to express FOXP3 and have T regulatory cell activity. And actually they didn't make anything like granzyme or IL-2 or other things that are more traditionally associated with cytotoxic T cell responses. And this is just the data here to show you that yes, these cells are associated with progressive disease, but also kind of that paradoxical thing where they're also positively associated with low levels of neurotoxicity. On the other side of the equation, besides running a bunch of bulk cell assays and qPCR for CAR T cell levels and TCR bulk TCR sequencing, we also ran what I called the 10x sequencing, single cell sequencing platinum package on all these patient samples. And so when I say platinum package, it's the combo of RNA, single cell RNA sequencing, single cell TCR repertoire sequencing, and we put a CytSeq panel together that was almost as deep as the CyTOF panel we were running, because we didn't want to miss a thing. And again, remember that these samples, we were running all of these samples fresh. So unlike the CyTOF work or some of the other assays where we were able to work from banked PBMCs that we thought at our leisure during regular working hours, this was all run and had to be chromium prepped in real time. And then we could let this library sit in the fridge. But this was something that was a quite extensive workflow. And the long story short is we weren't actually able to independently mine our predictive signature out of this data set. We were able to confirm some things about these cells. So what we did find is when we targeted these cells in this data set, it did show that these cells had gene expression patterns associated with the regulatory activity we had predicted. The other interesting thing that this data set did show is that responsive patients actually do have a polyclonal expansion, right? So it's not one CAR T cell clone that seems to be doing the work, it's a community of CAR T cell clones doing the work. So that was an interesting bit. I don't know if we didn't really need single cell data to solve that, to answer that question, but that question could be answered with it. But the point I want to make here is when you're thinking about setting these things up and budgeting, as cool as this stuff can be when we read about it in papers, it's something that is really hard to implement in practice. And this platinum package, by and large, took up more than 90% of our total assay budget. So it can be useful, but I think some of the things I was previously describing, and again, Shirley's presentation before this showed you don't even need, I mean, I'm not telling you don't do single cell analyses, because you should, but you can do a lot even without single cell data. And if you do bulk data analysis, if you're talking dollars per data point, it can be a lot cheaper. So with that said, there's a big group of people at Stanford that I get to work with. Obviously, I think I'm here because we're hoping to continue partnerships with the different oncology working groups and get new trials working with CMAC. Even if you guys don't work directly with CMAC, the slides I showed you at the beginning, the CMAC homepage, I forgot to mention this earlier, but all of the biospecimen collection protocols, standardized validated assay protocols, everything that they have painstakingly made us harmonize and optimize and develop. It's all on the website. It's all in the format that everyone wants to see inserted into your clinical trials protocols. If you need a biospecimen collection guide, we have one up there with all of the collection specification criteria for the different assays. And so even if you're not working directly with us, it is absolutely a resource that we've gone and run with and used for non-NCI related things as well. So that said, if there's a few minutes, I can take some questions, and if not, thanks for letting me rant for a little bit.
Thank you very much, Sean. I'm gonna kick off the questions while we wait to see if any of the audience, either here or on Zoom, has any questions. So I guess my first question goes to, if you're thinking about CyTOF versus MIBI, in terms of, I guess, throughput. You made the case for the fact that you can have bank specimens that you can analyze with CyTOF, MIBI, you can do on FFPE. So these are both scenarios that could be built into clinical trials and studies and specimens that could be collected. So is there, I know that some of the data analysis for MIBI could be quite intensive, right? And just wondering what your thoughts are in terms of throughput, or if you have any advice.
No, that's a really good question. So I think CyTOF has been around longer-ish. I was there in the dark days of CyTOF. I was, I don't want to say I was the first person working on it, but I was sure I can name all the people and count them on two hands. So it was a small group of us, and we've been working on it since 2007, 2008. And so the technology's had a lot more to mature, but with between sample barcoding and processing and a lot of the kind of published pipelines for going from like FCS files off the machine to data clusters, it's my lab with one instrument. We routinely run, 40, 80 samples a day. And you can do, generate a lot of those types of figures and cluster maps fairly routinely now. The machine learning, signature discovery, classification stuff, that's still a little more sophisticated. That's where you've got to work with your biostatistician. But getting things into a format that your data scientist or biostatistician can work with, that has definitely become more and more routine with CyTOF. With MIBI, but I think actually with MIBI, it's not just MIBI. I would say we are now being inundated with spatial technologies, right? Who hasn't gotten their email from NanoString or 10X or someone out there lately about their new omic box that you've got to buy that they can't show you yet because they're not shipping until next year? But it's like these things come with new data problems, new questions. I can tell you certainly with we actually just ran a workshop at the end of the summer to try and address some of this stuff. And so our lab is slowly but surely trying to come up with more standardized computational workflows where we can at least get people to normalize data and segmented cells from images. And I think we're there now. And the packages we put together work with both multiplex fluorescence data as well as the different mass spec imaging data sets. But it's still not as user-friendly as some of the CyTOF stuff. You still got to be able to have someone who can run some codes or some scripts. Also, in terms of throughput, none of these imaging platforms are fast. We run most of our studies on tissue microarrays when we can, but if not, running, if I do a study and I actually want to scan like a square centimeter of tissue, like basically the whole usable area of a slide, that takes me like a day. Now, 8 years ago, using the same assay with the old instrumentation, I couldn't even say it would take, I don't even know how long it would take. It would have taken longer than I could imagine. So things have gotten a lot faster, but you still have this imaging budget you have to work with, and all of these platforms are somewhat like that. So having tissue microarrays to go in can be helpful, but also just kind of switching slides can also cause limited amounts of time. But I would say with non-TMAs, the biggest study we've run now, I think we have like a 60 sample study that we're working on. And that took about a week to scan everything with people we using serial sections. We pre-selected areas we were interested in, and we had a couple of grad students coming and swapping slides. And starting with tissue microarrays, if you can actually work with your pathologist or your collaborators to pick and choose areas of interest and core those and put those all together in one study, which I highly recommend, because you might not want to run one assay, you might want to run different assays, and it lets you go back and know that you're imaging or analyzing the exact same area in the tissue over and over again. They can be super helpful. We've run studies we've run 1,000 patient studies doing that, because we've got all those patients on like 10 slides. And it took us maybe like a couple weeks to scan everything, but it's getting to the point now where it's like the wrangling the data has become more of a bottleneck than scanning the samples, which I guess is good. The idea is we got to keep pushing the bottleneck down. Yeah. And hopefully most of the work, which I don't know if you ever get past, comes to working with the people who understand these things biologically and clinically, where we actually get to just spend time staring at the data and saying, like, what makes sense? What feature do I want to partition and discover based on? What's the clinical feature I should be using to mine my data sets? So yes, I don't know if that's
that's very helpful. And I think the point about TMAs is very useful, because then you can really think of bigger data sets. Question? Yes, please.
Hi, Christine Ye from Multiple Myeloma Committee. We actually, we're very interested in those technologies, and Stanford certainly is the incubator of all those cool technologies. Two questions. I'm wondering, for the spatial technologies, you currently have imaging mass cytometry, you have Codex, you have MIVI. What are the differences between all these? And my second question is for the CyTOF, which is the liquid part. And you mentioned that you do store those samples, and did you notice any of those? Because usually, like a flow cytometry, fresh sample will be preferred. But if you store those samples, did you notice any viability causing any downstream analytical problems?
Yeah, no, they're all good questions. So we'll answer the last question first. So with the CyTOF work, or just, I would say, archival flow cytometry in general, I didn't make a big deal about it, but there's definitely a viability stain in there. You can have high levels of variability in cell viability depending on how long samples sat for before they were phycalled and frozen. And usually what we do, depending on what the assay is, we usually try to set like a viability cutoff. Like we'll often say something like, say, 80%. If the sample's less than 80% viable, then we really question the provenance of it, because maybe it wasn't handled properly before it was frozen. But usually working with these national biobank centers, working with centers who do this stuff regularly, you should be getting well above 90% viability on most samples when you thaw them. And one of the nice things with the CyTOF is you don't get autofluorescence, and that's one of the main reasons you usually try to get rid of dead cells, because they're naturally autofluorescent, but actually a lot of the time they still stain fine. But you should definitely incorporate a viability stain, which for the mass cytometry, there's a standard metal-based one that we use, but also for most flow-based assays, there's different covalent cell permeabilization dyes that you can use as well, zombie dyes and stuff from BioLegend. I forget what BD's is called. Back to the first question. How do I tell the difference between all these tissue imaging platforms anyways? And the tongue-in-cheek answer is there is no difference, right? They're all trying to measure the same stuff, right? So Codex, MIVI, imaging mass cytometry, they're all using antibodies with different reporters on them. So these assays are all using the same or similar antibodies to probe same or similar tissue and image and ask questions about it. So where the differences really lie is, one, which one do you have access to? I think that's important. What's the throughput? So I can tell you that MIVI has a much higher throughput than imaging mass cytometry, which is one of the, we're biased because we invented it, and we have a lot more infrastructure in my lab for it, but it also enables us to do, even though I was complaining that it still could be slow, it's still a lot faster than the laser ablation based work. Codex, Codex, I think the issue, the challenge you can have with Codex is it's still a fluorescent technology. So even though it's a lower plex fluorescent technology because you're cycling to get multiplexing, you still run up against immunofluorescence problems, right? For instance, you're never going to see someone do a Codex study on melanoma because you just don't, like I can tell you, even the CMAC stuff, the multiplex immunofluorescence groups that are doing that for their IHC stuff, we can't do melanoma because the weird pigments in the tissue make it too challenging, or sometimes you get weird pigmented gut tissue, and you know what I'm talking about. So I think you also have to think about the matrix that you're trying to analyze and the type of sample. And then the other thing is there's also sort of a data quality, resolution, sensitivity thing. I think we haven't done direct benchmarking, but based on first principles, MIBI is a more sensitive assay based on how it works, but there might be molecules that you might have trouble measuring with these other assays, but at the same time, the hardware for MIBI is the most expensive out of the three. So it's like a multi-dimensional matrix of optimization. I can say the one you have access to with people who can support you using it is going to be the best one to start with. But I would definitely take a close look at, if you're considering using one of them, take a close look at whether or not the first study you want to run, how compatible is it really with that tech? Can you run a pilot study with the company or run one sample? Can you get the reagents to support the biological features you want to measure? And yeah, that's really it, if that makes sense.
It does.
Yeah.
So you mentioned the CMAC, like your network.
Yep.
And you're up for the grant renew. So is that open for the trials? Because we'll be launching a study, and we actually plan to incorporate immune monitoring into the study. So how can we kind of like consider that as a resource?
Oh, you should, if it's an so the default for CMAC is phase 1, phase 2 NCI sponsored trials. Even outside of that definition, there's other pots of money to pull from, and the language is vague, but in the CMAC renewal, they're actually considering even trials that fall outside of those conventional phase 1, phase 2 NCI definitions. So the idea is that, the contact info is on the website. If you don't get a response from them, you can email me directly, and I'll shuttle you towards Rebecca. But usually what happens is we have as with all of these things, we have lots of meetings, and usually the trial leads come and present at one of the meetings, and we have a discussion, ask questions, and we're like, okay, does one of the sites want to kind of sponsor and take the trial under their wing? And if one of the sites agrees, then usually what happens is, even though you get access to assays from all the sites, one site basically agrees to be your CMAC Sherpa, and we carry you through the pipeline, and we get all the assays and biospecimen amendments added to your protocol and approved. We do all the coordination with the biobanks to work with you to make sure samples are collected and labeled properly. And then we also make sure that the assays that we're running are best customized to help answer the questions in the trials. So that's, yeah. And so the CMAC network has been renewed. We don't all know as individual sites if we will be renewed, but it is one of those limited renewal things. There is a pot of money, and they're planning on paying for this, and they have goals to, get another something like, I want to say it's like 6,000, 8,000 patient samples through the network over the next 5 years. So we're definitely enrolling new trials. There might be a little bit of a lull right now because we're all going through the renewal, but the money is there. So I think everyone would be really sad if because we actually, as you might suspect, the network, there wasn't patient samples to run on day one. So we actually spent the first kind of half of the project period really just planning and validating, writing all these protocols. And it's really then finally samples started coming in, and that's when actually your 5 years is up and it's time to renew again. But I think that, it was the moonshot decided to, with all the other things that were refunded, the network there was also refunded. So you should absolutely, the IMATCH trial, I think I think all of the anything that you're doing, and even if it's not one of these, again, one of these NCI-sponsored trials, if it's a company-sponsored trial, if it's an investigator-initiated trial, as long as you don't have crazy criteria around data sharing, you could get resources and expertise from CMAC to do some of your biomarker discovery with.
Great, thank you so much.
Thank you. Thank you very much for that great presentation. And thank you everybody here in person and also virtually for attending and participating. Siwen, did you have anything that you'd like to say in closing?
Oh, I just want to thank everybody for coming to our committee meeting and for staying until the end. Thank you so much. Hope to meet you soon.
Thank you. Thank you, everybody. Have a great rest of the SWOG meeting.